Oh, and the guinea pig water. All right, and you're gonna have to go thirsty for a little bit. Okay. Got this, share screen, go to this. Alrighty, so we've got the reproductive system. Go to this fill the screen with it. Hopefully you got the whole screen that says reproductive system here. And then obviously there's just the male, female. The male will do the anatomy and physiology. We'll cover all of the male components today. And then that way we can have more time for the components of the female physiology, which can be a little bit more complicated because there's several phases involved. Okay, that's the whiny protest of the guinea pig if you hear a sound. So as far as the reproductive anatomy for the male, we have obviously the skin on the outside around the testes will be the scrotum. And it's within the testes that spermatogenesis occurs. So we're going to spend some time figuring out how sperm are actually developed. The duct system that's listed here really just means the pathway that sperm takes from where it's made in the testes all the way up into the body and then out. So then the urethra, we're going to recap that. Obviously, that's on the exit out. We already talked about that with regards to urine. It also plays obviously a role in the male reproductive anatomy, as well as accessory glands. These are glands that are going to actually create seminal fluid. The structure of the penis and what actually semen is composed of. So that's our topics for today. We have here a side view, obviously, of the male reproductive system, obviously kind of a cartoon version of it. And we can see one with more of the musculature and stuff involved and other elements, and then the one on the right is a little bit more paired off. But I wanted to point out to you again with the ureters being put in the wrong location. So I'm going to circle this here. That's a ureter, and it's arrived, they should have it drawn as coming in the top of the bladder, when really the ureter should come down and actually enter the bladder on the posterior, more on the inferior side. So I wanted to point out that inaccuracy. Sometimes people ask while we're in class what this is. That is the symphysis pubis of a bone, the bony structure, so of the pelvis. So that kind of gives you a frame of reference for that with regard to the skeletal system. So as far as what we have here is the testes. Obviously the testes or testicles are located outside of the body. They're within the scrotum, outside the torso, because the temperature for creating sperm needs to be slightly lower than body temperature. Not a lot lower, but just slightly lower than body temperature. So there are surrounding muscles right under the skin, known as the dartus muscle. There's several of them, but the dartus is the only one that we'll mention here. And that's actually what gives the skin a more wrinkled appearance. And so its role is actually to maintain a temperature balance for the testes. The tunica albuginea is actually a really tough serosa type layer around the testes themselves. And then the seminiferous tubules, these are the guys that we're going to spend a lot more time on, because that's exactly the location that sperm are actually being made within the testes. So we can see in here, the testes is this portion. What we're seeing on the outside, sort of this covering portion, is the tunica albuginea. And then we have, move the video, people, images are off to the side. And then I think that's all we have in this particular image. We also have, we can see the, here, the dartus muscle is the one that would actually be going around. In this portion, that would be, oh, the dartus isn't here. The spermatic cord is this portion. So the spermatic cord is really not only the vas deferens that's actually going to bring sperm upwards, known as the ductus deferens. That's going to be the path that sperm follows. But we have a lot of vessels and arteries, lymphatic vessels and nerves. So the whole spermatic cord itself means all of the stuff coming and going from the testes themselves. The surrounding muscles around the spermatic cord itself, though, that actually involves is the cremaster muscle, and that is more about retraction of the testes closer to the body. And then we have in this picture the tunica albuginea, which is the outer covering, and the seminiferous tubules are further inside. So here's a really cool old picture by Netter. He's a really famous anatomy artist. And so this is a really nice picture where we can see, you know, I just have them expanded. The dartus muscle would be out here where the cremaster is around the spermatic cord itself. We have the testes here, and then later we'll learn that this portion here is the epididymis. When someone slices open a testy, this is like a single testy here. And so notice all these little tubule networks inside. So when someone takes a slice, a histological slice, you're gonna see there's gonna be like through this here would be like making a microscope slide that we end up with here. So because there's so many tubules over here, we see these tubules coming out the screen towards you, but they're like, you know, lots of little like hoses on someone's garden. So you're just cutting right through it. So in the middle, in the lumen, that's this area, is actually where sperm will be. We can see sperm formed at this point. So that's where sperm are made within the wall. The start of sperm production starts from cells out in the surrounding wall of any one or all of these seminiferous tubules. So where I'm circling are these cells, or just sort of drawing cells in the outer portion of each of these seminiferous tubules. I'm going to just erase all this, or just do this. Okay, so sperms start to be made from cells on the outside. They continue to develop. They go through the process of meiosis to ultimately become a sperm here that sprouts a tail. Then it detaches from this inner wall, and then here the sperm with a little tail, and it will then migrate through the seminiferous tubules. We're going to go back to this here, goes through all the tubules, and then we end up in that epididymis, and then ultimately out, going to go up into the body. So here we'll look at a single seminiferous tubule. Again, these are tubules found within the testes that make sperm. The cartoon picture on the right kind of gives you a better depiction of the cells. We're going to want to look inside the cells, so we want to see this a little more clearly. You can see it's how difficult it can be with the slide to the left. But I want you to notice that the cells look more, I guess, normal, cellness out here, where notice in here, they're kind of black and squiggly lines inside. That is because the chromosomes are unwound. So in a normal cell, you have, you know, we always draw a cell, and then we have like a really dark nucleus. And so I'm just coloring it, and then that's a cell. Well, the nucleus is so dark because, let me just do the stop screen share for a second. So the nucleus is really dark because the stringy chromosomes and the DNA and all that is wrapped around. So it would be like if I don't have string in my office, but if you had some yarn and you had this giant pile of yarn, and in order to pack it down, maybe I took, you know, this pen and I'd wrap the yarn around it, and I could get a big giant pile of loose string packed up pretty tight if I wind it and spool it up around things. So what a nucleus is all of these chromosomes packed in so tightly and compressed down. So when you're undergoing meiosis, then you sort of let them out. And so the pictures where it shows the cells, instead of with nice, normal, tight, packed in little nuclei, but like squiggly lines for nuclei, then you know it's in the process of meiosis. So let's go back to this image. So we can see that these cells, they started out over here, and now they're moving their way inward as they start to uncoil and release they're chromosomes and they're kind of squiggled around. It's undergoing the meiotic process, but then ultimately it recondenses down, and then you have another back to being tightly packaged, and then you sprout a tail. So that's the process in a nutshell, but we are going to go through the specific cells in order that this is going to take place. So of the seminiferous tubule, we have sperm-producing cells in here. But outside these seminiferous tubules, we have cells that are known as Leydig cells or interstitial cells. But these cells make testosterone. So obviously testosterone can feed back into the cycle of forming sperm, but it also obviously has secondary male characteristics where it increases muscle mass, increases vocal cord thickness, which decreases the voice, the tone of the voice, as well as, you know, hair in the face and things like that. So those are, testosterone does things throughout the whole body as well as contribute to sperm production. So again, inside the seminiferous tubules, here's some adjacent area here. That's going to be the Leydig cells or interstitial cells, where the sperm producing cells are here inside the seminiferous tubules. So this is the master list of the cells that you're going to need to know. You will definitely be getting a slide image of a seminiferous tubule. You will definitely need to know and be able to label each one of these on the slide, and you'll need to know what they do or what their role is. So this is sort of the master list, but we'll talk about each of these with the picture in mind. But I just wanted to point this out. So we'll use these pictures as we go through that list of cells. So in a single seminiferous tubule, the cells out here in the periphery, so let me just sort of kind of that area, and we have our cells here, they are known as spermatogonia. Think of that as the daddy cell, the daddy of all the sperm cells. A spermatogonia has normal 46 chromosomes. That's what a normal cell has. We have, or you can call it 23 pair of chromosomes. So our 23 pair of chromosomes, which happens, just double it, which means 46. That's what this start cell does. It's the beginning cell. It is this cell, each one of these, that will go through meiosis. So a single spermatogonia is going to then undergo the meiosis process to ultimately make 4 tiny little sperm cells. That's what we're going to end up with. So you start with a spermatogonia at 46 chromosomes, but you're going to end up with a spermatid or spermatozoa with 23 chromosomes. Ultimately, the 23 chromosomes from a sperm are going to combine with the 23 chromosomes in an egg, and then you're back to a complete cell or a complete set of 46. So I want you to keep in mind, because you're going to need to know on each of the cells listed here what their chromosome number is. So we already know that spermatogonia has 46. Also, we can say 23 pair. That's the same thing. The primary spermatocytes, we're going to go through the, I also wanted to show you, this is would be like a normal nucleus, but then you can see with all the stringy stuff out, these are at different phases. This is actually a plant cell, I'm pretty sure, at different phases of the actual mitosis process. But we're going through meiosis, but I really wanted to show you how these, you know, that packed up nice and tight versus all loose and stringy. So when we get into, I'm going to go back 2 slides, into these primary and secondary spermatocytes, those guys are the stringy ones. They have already unraveled. We're undergoing the chromosome. This is where I say you can see the chromosomes inside. So they are the stringy looking ones. So they're easy to spot because of that. While we have this here, we're still at 46 at this point, but we come 23 chromosomes at this point. And then it stays 23 as we go on. These last 2 cells here are regular cells, and we'll talk about their role. We already mentioned interstitial cells, also known as Leydig cells. and they all are normal cells, so they have 46 chromosomes. So the only ones that have 23 are the ones in the latter stages of meiosis. So we have spermatogonia, you can see that here, and then we'll have, let me see if there's a better picture, but we see primary spermatocytes, so you can start to see the cells are more speckly inside, and then a little further inward, you'll probably have a secondary spermatocyte. And so notice these guys, any one of these here, a primary or secondary spermatocyte, same with these guys. Generally, you really can't visually, at least at our point, tell the difference between a primary and secondary. Usually just a primary is further out here, or a secondary is just that much closer to the lumen. But they really look pretty much the same. So based on the positioning of this primary spermatocyte, we can definitely call this one a secondary. And actually, if you put it's my 2 with a 0, it looks like 2 degrees. The 0 at the top is shorthand for secondary. Primary shorthand for that is that. Anyways, that's from chemistry. So that's your primary spermatocyte and your secondary. But then look at how this cell, all of a sudden, the nucleus is starting to recondense. That cell, I'm going to go back to our master list. Let's erase all this. That cell is going to be a spermatid. So let me go back to the picture. Oops. So this here is a spermatid. That means we've condensed back down the nucleus, everything's packed back in, but it does not have a tail. So we'll go back here to this other thing. And so then the spermatozoa, that's here. So let me make this one, since I put a square. This one's a spermatid, and these guys are spermatozoa. You can kind of ignore what this is labeled because it says spermatid maturation. We'll just name it a spermatozoa because then that's really close to a mature one. The spermatozoa is technically after it detaches, but let's make that the distinction here. We either have a solid nucleus with a spermatid, and then this is where this nucleus even condenses further down, gets packaged even smaller, and then ultimately a tail will sprout. So that is the process. So this is what meiosis really looks like, where we have 46 chromosomes, 2n, that means 2 times 23, the n is 23. So it means you have 2 sets of 23. Then we get into the sort of primary spermatocytes level, and then it divides. And then when it divides, we actually have 2 23s. They then replicate and become 23 chromosomes. So ultimately, we can see here for the male on the left side of your screen, this is the division. So we really start with a spermatogonia at 46 chromosomes, and then we ultimately get down to spermatozoa at 23 chromosomes. The female, the process, you end up with the 4, but the way it goes, the cellular substances and components really favor a single egg. So you end up with these 3 kind of discarded components, and they all are going to have 23 chromosomes in it, but there's not enough cellular substance in these polar bodies to make them functional. So in the female reproductive system, the meiosis process is similar. It's just that the cell itself robs from the other cells to make one giant master cell, if you will. So the males are pretty much easier to deal with when we talk about meiosis, because you start with 46, you end up with 23 same identical little guys. Well, not quite identical, but with regard to 2 of them, these 2 are and those 2 are.
I have a quick question. Will we need to know that like exact slide to be able to label that?
Which slide? This one? No.
Yeah.
Not this one. This is more just to let you know the process of going from 46 to 23.
Okay, thank you.
You're welcome. So here we have, I think, what am I, oh, I had this schematic. I made this last year, so this is sort of new. I forgot, I've only used this in class once. So we have the spermatogonia, which is going to be out in the periphery. Then we have primary spermatocytes, and you can see them with the wiggly chromosomes in the, inside. Then we move down to secondary spermatocytes. They still look really similar. They're just sometimes kind of embedded with it, just slightly closer to the lumen. Then we end up with spermatids where the nucleus is starting to get solid again. It's still a little bigger cell than normal, or than it would be if it was a sperm, because finally the spermatozoa, the nucleus shrinks even further, and then ultimately a tail develops. And finally, these guys will then detach from the wall and start to travel through the tubules. So these are the four main or five main cells that you need to know, and you should, and this slide here has all the chromosomes that they are, the level they are at. Then when I sort of did this first, so I put a couple of these other seminiferous tubules like over here and over here, just so we don't have to go through the cells of them, but then to show you the spaces in between, we'll do these Leydig cells. So the Leydig cells, we already talked about them, known as interstitial cells. They are the ones that make testosterone. Who remembers what hormone from the pituitary gland targets Leydig cells that stimulate testosterone production? Okay, the answer is the luteinizing hormone does that. So luteinizing hormone from the pituitary targets interstitial cells, tells the male to make testosterone. While we're talking about the pituitary gland, follicle stimulating hormone is going to target spermatogonia to ultimately go through the sperm production process. You have to know that. There's a slide coming up about it. I just thought about it here. So then I threw in these crazy triangle cells. These triangle cells are known as Sertoli cells. Another name for them is sustanacular cells. These guys have 46 chromosomes. They're the same as a normal cell. They're within the seminiferous tubules, in here, but they actually don't make sperm. They're not in the process where they're going to ultimately become sperm. But what they do is, I'm just going to change the color, do a little green color. They actually provide some nourishment that actually fuels and helps these cells that's going to, these guys are going to become sperm. But the Sertoli cells, you want to think of it as feeding or providing sustenance to these cells. So it's facilitating the production of sperm. It just isn't going to be the production of sperm. And so they're listed here as a triangle. I kind of think of one way when I was a student and I tried to remember Sertoli cells, and I've got all these other names of cells I'm supposed to remember. So I always thought Sertoli sounded kind of Italian, and I thought, oh, they always draw them as like these little triangle things. So I always picture them as like a little piece of pizza. It's not going to be the sperm, but it feeds the process. So that was, anyways, it's quite goofy. So you can just ignore that if that doesn't work for you. This slide here is not something that you will be tested on. It is just something that I'm showing you of how everything relates. You've got DNA way down here, and you can see how DNA is wrapped, sort of beads on a string, so it's sort of wrapped around here. Then these strings get looped around, ultimately coiled. You can see this whole packaging of these chromatins. Here's another slide here. We have the DNA, they're bundled up. We link all the bundles together, so we package them even better. And all of that is in just one section of a chromosome, and the chromosome's extended. So then when you package up a chromosome, then you can see how much is actually packaged into one little area. So it is really remarkable how these things are put together inside our nucleus. So this is not to be tested on, this is just to show you. This is another picture. I just thought it was quite a cool picture where you can see how things are packaged, where we go ultimately from DNA and then down to ultimately like the whole chromosome that we're talking about here. So get out of here. Okay. I keep having to move the menu I'm moving it down, moving it up, for the screen share. So this is a view, again, we can see our spermatogonia cells, our primary, secondary, we have our spermatids, and ultimately our spermatozoa. we can see our Sertoli cells within here, and we see our interstitial and Leydig cells that make testosterone outside here. This is a histology slide, so we can see this one's just numbered, so you can look at this one and enlarge, but we'll continue on. So I think but just know that you need to know the process and the name of each of these cells. You can see a nice Sertoli cell. Okay, so on to actually the anatomy of a spermatozoa, called sperm for short. So we have the acrosome. Let's see, let me get this back to red. I always think the acrosome seems better. The acrosome is like this little cap over the sperm itself, and it has these digestive enzymes. And what's important about these digestive enzymes is that this is how the sperm will ultimately penetrate the egg. So the acrosome must discharge on the surface of the egg to allow it to penetrate it ultimately. So I'm going to switch slides here, switch colors, it's blue. So then the head of it is actually within here where the nucleus is. And so the acrosome is like a little cap on there. Then we have the mid piece, and within the mid piece is this mitochondria. Well, why we need mitochondria is because this is really our motor. This is moving side to side, and when it moves, it causes the tail to actually move. So the tail is really like a whip. So let me say like a whip. A whip would just lay there. It's not going to move at all until somebody picks it up. The movement of a whip has to be started from someone's hand and the flick of a wrist, which is what the whole mid-piece's job is. So the mitochondria, the energy production here, is actually causing the movement. So the movement's occurring here, but then as a whip, it extends out, and that's ultimately how the sperm will swim forward. But what's important is mitochondria, when they actually create ATP and do this movement action, it's like a nuclear power plant. When you make energy, you actually can create free radicals and some other damaging substances. So you do not want to activate sperm until you're ready for it to go, because once sperm becomes activated, the mitochondria start moving, then the life duration of the sperm is severely reduced, and it will only last anywhere from 2 to 5 days. So the whole process that it goes through the male reproductive system, from the point that it's made in the testes, and then until we're going to go through the duct system to where it's ready to launch, the sperm are not moved by the mitochondria. They are in a very halted and inert state. The mitochondria only gets activated at the moment of ejaculation in combination with some of the other seminal fluids. So we're going to go through the parts of the duct system, the pathway. So we should really say, starting from the Seminiferous Tubules, then it will end up going to the epididymis, and so this is the rest of the pathway out. And then along the way, we'll talk about the glands that are actually going to form and contribute to seminal fluid. But we'll go through the ducts, which is really just, again, the sperm's pathway. So we have the testes, obviously down here, the testes, and then this portion at the top, and actually arches around to the bottom, is known as the epididymis. I think we have that labeled here. So if this is the testes with the seminiferous tubules inside, then let me change colors. Then the epididymis is going to be this portion. So it's this whole portion right here on the outside. So that's the epididymis, and it is just continued set of tubules all through here, and it is during this time that the sperm actually mature. So they're not using their tail. It was just, they were made in the seminiferous tubules, and then this is sort of their maturation. So we have additional tubules, so it takes them a long time to get through here. So we have additional tubules for the sperm to travel through, and then finally you get to the bottom of the epididymis, and then see where here, where there's no more wiggly tubules, then that's going to be the vas deferens. So the epididymis is only this portion where we still have wiggly tubules and it's cupping around that testy. So the vas deferens, also known as the ductus deferens, is this part here. This is the part as it's going up into the body. But I want to point out is look at how thick it is. The wall of this vas deferens is incredibly thick, meaning the lumen size where sperm goes is right here in the middle. It is so tiny. Just to give you reference, what we have here is a vein and an artery right next to it. So those are teeny tiny veins and arteries. I'm calling this one a vein because you can see how thin walled it is. And then you can see how this one's a little thicker compared to this other one. And it's an artery. And it has higher pressure, so it's actually recoiled in. Obviously, this is from somebody that's removed from the body, so it's actually retracted. So veins stay bigger than arteries do after death. So the vas deferens, this is smooth muscle. And the smooth muscle is important going up here into the or up into the body because we don't want sperm to move. It's not moving with its own little tail. So we have peristalsis that's actually occurring that's literally moving sperm up into the body like a little conveyor belt. So the sperm doesn't have to have any effort on their part. So it's going up anteriorly into the pelvic cavity, but then it goes up and then behind. So it's actually arching around and going behind the bladder. And finally, it gets to the end of the vas deferens is known as the ampulla. So we'll go this. So this here is the ampulla. And it is the end of the vas deferens. Let me see if I have another picture here. So the ureters are bad in this one. This is the bladder, obviously, but this is the back side of the bladder. It's the posterior side. So going back over here, we can see my picture. This here is obviously the bladder. And then, I don't know if you can see very well, but this is going up in the body, but then it's showing you kind of see through how it goes behind the bladder and you end up in a wide spot known as the ampulla, but again, that's on the back side. So when we go here, move my picture again, whoops. Sorry, the video pictures are in my way every time I go. So this is the posterior side of the bladder. So this here is the vas deferens. And then I'm gonna put a little A, that's the ampulla. So the ampulla of that. So you can see where it's wider. So it's like a little storage area for sperm. And so once sperm are made in the seminiferous tubules in the testes, they migrate up through the epididymis, they go up into the body, over and behind the bladder here, and they hang out in the ampulla until they leave the body. So sperm sits here quiet as a mouse. They do not move. They are inactive. We don't want that midpiece to move. We want to protect the nucleus at all costs because it is so streamlined. There's really nothing to protect the nucleus if any free radicals are near it. So the sperm has to stay very, very still. So it would just stay there. It stays there for a couple of months. And after about 3 months, the body just sort of reabsorbs it if it doesn't get used or released or however you want to rephrase that. So, and sperm production is very much a use it or lose it situation, maybe not lose it, but at the same time, if people are men that utilize or release it on a fairly regular basis, increase their ultimate production in the testes. If sperm hangs out in this area here for a while, then there's a feedback mechanism that goes back to the testes to say, whoa, hey, we're full up here in the ampulla. We don't need any more. We're not releasing any right now, so let's just slow down production. So that's where the storage is. So this is before ejaculation. This is where it hangs out. So then notice these really globular glands next to the ampulla. Those are the seminal vesicles. those are the largest secretions that we will receive in ultimately what forms semen. So we keep the secretions separate from the ampulla. You know, the sperm are hanging out in the ampulla, and then we have these seminal fluids, or the glands for them, sitting next to it. They will only combine at the moment of ejaculation. But while the sperm is at rest and when you're not releasing sperm, we don't let them mix up. And it's because the secretions from this gland is what's ultimately going to cause the midpiece of the sperm to become activated. So we know the epididymis is where sperm are matured. We know the ductus or vas deferens brings the sperm up into the body, up over the bladder, and at the very end of the vas deferens where sperm is stored is called the ampulla. At the moment of ejaculation, we're going to go the rest of the way out. It's going to go through the ejaculatory duct, which is going to be in the prostate, and then we go through the 3 levels of the urethra. So we start off here in the ampulla. This is where sperm hangs out. We have seminal vesicles here on the side. At the moment of ejaculation, there is a little channel coming from both sides where fluid from the seminal vesicles combine with the sperm in the ampulla. They combine together in the ejaculatory duct. So hopefully I have a different picture of it, because you can't see the ejaculatory duct because it is inside the prostate. So hopefully, yeah. And also keep noticing the ureters are still put in the wrong place. The ureters are always placed really high. So these, again, are really inaccurate drawings where the ureter should be entering a lot further down. Next picture also does the same thing. It's got these really badly drawn ureters. Unless they've got the bladder really tipped up and forward, it should be coming in down here, closer to the floor of the bladder. Okay, so this here is the vas deferens. This here is the ampulla. These here is the seminal vesicles. Coming into the prostate, each ampulla seminal vesicle has its own ejaculatory duct. So we have fluid from the seminal vesicle as well as fluid from the prostate gland that will contribute to semen. This is the best picture here. So we can see this is a side view. And so this here would be a seminal vesicle. We have that. And then we'll just sort of draw an ampulla, put a little A here. So the two together, this itself is the ejaculatory duct. So you can see it's actually a little tunnel through the prostate, this being the prostate, and it's ultimately meeting with the prostatic urethra. So if that's the prostatic urethra in red, then maybe I'll change colors here, go to blue. This here would be the membranous urethra, and then you would go out, obviously, the penile urethra. So then I'll change colors again. I'll do purple. The ejaculatory duct in this case is the purple here, and so it enters the prostatic urethra, and then ultimately the sperm and the seminal fluid from the seminal vesicle and fluid from the prostate gland will then go into the urethras and go out the body. Okay, so here we have this picture again. We see testes down here. Maybe I'll do a yellow color here. That might show it better in this picture. Okay, so we have testes, then we have our epididymis, then we have the vas deferens going up. Now here's our vas deferens coming in. We come back, and now it hangs out here in the ampulla. So I'll widen here. So now we're at the ampulla, and then next to the ampulla is going to be a seminal vesicle. They then, at the point of ejaculation, come in through the ejaculatory duct. It's going to meet up with the prostatic urethra right here. Then we have membranous urethra, I'm drawing with an X, and then squiggle out through the penile urethra. Okay, so we already know the urethra, prostatic, membranous, penile, or spongy. It's also called spongy too. We'll get to that. We have sphincter muscles within the prostate because it's sort of like I was thinking like a train track switching station. The sphincter muscles located in here will seal off the bladder so that we can let semen go, or seals off here so we can let urine go out. So that is, we actually have sphincter muscles that prioritize whatever's needing to go out at any given moment. So the seminal vesicles will go specifically of what they've got in them. So we already talked about how seminal vesicles are located here. It's going to be the majority of seminal fluid, of ultimately what is contributing to semen. What does it have in it? Water, so that gives it the fluid component. Fructose, well, fructose can actually is like glucose, where it's what's providing energy to the mitochondria. So this is where the mitochondria get activated. Prostaglandins, that is a local hormone. What do we need a local hormone for? Well, not for the males. What's going is in the seminal fluid. Where this is going is ultimately, if we deal with biology, to the female reproductive tract. In the female reproductive tract, the prostaglandins, the goal for them is to actually soften the cervix of the female, which is the gateway into the uterus. And so it's actually trying to open that up so sperm can get into the female reproductive tract more easily. So that's the role of prostaglandins. And then fibrinogen. Well, we have fibrinogen we learned about that when we did with blood clotting. That's ultimately what prothrombin turns into thrombin, then thrombin turns fibrinogen into fibrin, which is going to actually be the fibers that makes a blood clot. So you're like, what is this doing in seminal vesicles? The fibrinogen help it to stick. It actually causes it to blob and glob up. And you actually want that to happen in the female reproductive tract. You're actually creating this sort of bubble area. Not a bubble, not like with air, but you're actually creating this blob of semen in the female reproductive tract that's going to be just adjacent to the cervix to actually make it easier for sperm to swim into the female reproductive system. So each of these things have a job to do. In the prostate, obviously it's below the bladder, it has a prostatic urethra. It contributes about 20 to 30% of semen. It has additional components that facilitate sperm motility. That means moving the tail around, that's getting the mitochondria to move. It also is more alkaline. And the vagina of the female reproductive tract is really acidic. And acidity can cause damage to sperm. So when sperm is delivered to the female reproductive tract, it has to have these prostatic secretions that actually buffer the acidity so it doesn't harm sperm. So here's a recap. You should be comfortable labeling anything on here and describing what it does for your exam. The last of the glands, there are 3 glands. We talked about seminal vesicles and we talked about the prostate gland. Those two provide the majority of the fluid that's contributed to semen besides the sperm. Cowper's glands is actually located right here. So you can see this little guy. So we've talked about the seminal vesicle there, and we've talked about the prostate there. And the third gland is known as the bulbourethral gland. There's actually 2 of them, but in this side view, we just see one, also known as the Cowper's glands. It's at the base of the penis. And so what they do, they're under the prostate, but at the base of the, where does it say? Below the prostate, should be at the base of the penis. Between the penis and the prostate are these Cowper's or bulbourethral glands. So they mean the same thing. They secrete an alkaline fluid. Again, I'm trying to protect sperm, but their first fluid secretion is really neutralizing any remaining urine in the urethra. And then it actually lubricates the inside of the urethra for the male so that when ejaculation takes place, things can roll on out a lot more easily, and it provides early lubrication. So this Cowper's glands or bulbourethral gland is really the first one that causes secretions out the penis. Then at the time of ejaculation, it's really about the seminal vesicle and the prostate and the sperm here in the ampulla that then gets delivered together. Okay. This is a frontal slice. So we have the bladder up here. I wanted to point out the urethras are now, the holes there are shown appropriately. And here is the, ureter. Holes for the ureters are shown here and the single urethra there. So let's just recap this here. Remember, it's the trigone. You also can see the rugae around. This is the only part of the bladder that doesn't have the rugae. It stays nice and flat there. Then we have the prostate. I should probably change colors. Okay, so here's the prostate. We can actually see lots of little holes in here where the secretions of the prostate can come into that prostatic urethra. And then we have the bulbourethral glands here. They then, this here is the base of the penis at this point here. Okay. So what is semen? It is seminal fluid plus the sperm. So what is mostly water? Semen's mostly water and some sperm. So sperm is less than you would think within semen, but the semen is providing the fluid and the mechanism for sperm to make it into the female reproductive tract. It provides something alkaline to protect the sperm from the acidity of the female. It actually facilitates motility of the sperm, meaning activate those little tails so it could move. It has the prostaglandins that's going to help make the female reproductive tract a little more accepting of sperm so it can actually make its way in. And then the fructose for energy. So finally, the penis, what are we going to do about that? It's urine and semen transport here. So in the penis, here's most men don't like this. If you cut a penis right across the way, although that's what it looks like. Now, I'm trying not to be really immature, but it does look like a face, so sorry. And on this, if we have the two areas here, these are dorsal arteries, these little red circles there, and they are actually the main blood flow into the penis. Now the tissue around it here, that's sort of around the eyes, if you want to be immature like me and see it as a face, that's known as corpora cavernosa. So how an erection takes place is blood flow comes in, but then fluid from the blood radiates out into this cavernosa space, and that's actually ultimately what inflates. And what inflates with blood, and that's ultimately what creates the erection. Now notice we have corpora spongiosum down here that's sort of surrounding the mouth area. Again, this area right here that seems like it looks like the mouth, that is the urethra. That is the penile urethra itself. So the point of corpora spongiosum is actually to inflate, actually gets its erectile tissue, but it does it to keep the urethra propped open. It does it to prevent excessive fluid and blood filling the cavernosa. You don't want it to encroach and block or pinch off the urethra. So the corpora spongiosum also inflates around the urethra to protect it so that that pathway out can remain open. So in summary, the penis is used for both urine and semen transport. It has the main erectile tissues are the two corpora cavernosa tissues that are on the dorsal surface, and then the corpora spongiosum surrounds the urethra. That's why this part of the urethra is known as the penile urethra or the spongy urethra. So either term is applicable. This picture on the right is a really great picture because not only do we see the bladder, we see the trigone, we have kind of a see-through bladder where we can see the ampulla in the back, and we see seminal vesicles next to it. You don't see ejaculatory duct, we see our prostate. We see our bulbourethral glands here. So this is a really nice picture, shows a lot from this particular view. I forgot to even say the testes and the epididymis and the vas deferens. Here's another picture. This is an actual histological slide of a penis. I think it's a rat penis is what we usually have some in our lab. they're small, and I think they have rat penises, is what you can see. The drawing is probably a little bit better, where you can see these dorsal arteries. You can see the cavernosa tissue here, as well as the main dorsal vein there. So what is an erection? Well, obviously not what is it. How does it happen? Arterial dilation. So during a parasympathetic, this is where like you got the Barry White going on, mellow, parasympathetic, you're actually causing arterial dilation. So you're increasing blood flow there. And so then you have smooth muscle relaxation. We're facilitating blood flow. There's actually a corresponding occlusion on the venous side. So you're letting blood in, but you're preventing it from going out. So basically it's inflating. And so where is this fluid going? It's going into the cavernosa space. So it's actually the more blood that goes in, this outer tunica albuginea, and we talked about that around the testes, there's also a layer around the penis too, it's actually causing it to compress venous outflow. So again, blood flow is going in, but we're preventing it from going back out. But it's important to have this parasympathetic activation. People with erectile dysfunction often happens later in life. It's not always a lack of function of the penis. It's often lack of not getting in a parasympathetic mode. People get stressed out, their brain's thinking about maybe a million things, and maybe they're thinking about performance issues, whatever, but you got to get parasympathetic. That's the beginning. And the reason why I make a big deal about parasympathetic is because actually at the point of ejaculation has to be a very quick switch over to sympathetic. So the whole act of sex requires both parts of your autonomic nervous system. You need the parasympathetic for the buildup, but then the ultimate release and the orgasm itself is a sympathetic nervous system action, which is why when there's a sympathetic action, there often can be a corresponding deflation. So here's a better picture. You can actually see some of the tunica albuginea. This actually came from a research paper about the process. So if you can read more about it if you want. So I mentioned these hormonal regulation before. From the pituitary gland, remember we had follicle stimulating hormone, FSH. FSH is going to make sperm. Luteinizing hormone is going to make testosterone. So I want you to be really clear. When we have seminiferous tubules, the Leydig cells out here are stimulated by luteinizing hormone. Inside that makes sperm, follicle stimulating hormone is going to be targeting that. Okay, so I think I must have a little schematic here. I forgot about this slide. Okay, so the anterior pituitary system is going to send out follicle stimulating hormone that will target spermatogonia within the seminiferous tubules. The action of that would be stimulating the formation of sperm through the process, through the wall of these seminiferous tubules. On the right side, we see the anterior pituitary is going to release luteinizing hormone, and that will go to the cells in between seminiferous tubules, known as interstitial cells or Leydig cells. The job of those guys are to make testosterone. So I want to be really clear that you understand the role of both of those hormones. And I'm going to stop sharing. Okay, that's it. So I think we've been having a lot longer lectures, so I wanted to give you a treat and shorten one up here for you. So the male reproductive system is short and sweet compared to some of the other stuff that we have to do. Does anybody have any questions? Okay, so that's it for today. When I see you on the next lecture, we will start covering female reproductive system. We'll go through female anatomy, and we'll probably do the basic outlines of some of the female physiology, and then we'll stop, and then we'll go on to more of a calendar thing and more female physiology and histology, and then get into early embryo development. And then that'll take us to the end of this unit. Okay. Let me know if you have any questions. You can sign up for student hours on the Calendly app that's on our Canvas page, and or just shoot me an email. I'm happy to help in any way I can.